Hello friends, welcome to another session of Biochemistry. I think there was some confusion regarding the timing of the class, whether we have a class today or not. So we are starting so late. So we'll have a 1 hour session today, and then we'll finish the biochemistry, microbiology, and whatever is left tomorrow. So just let me know you're here. Give me a hi. Let me know if you can hear the audio. Let me know if the video is clear, and let me know if you can see my iPad. Hello and good evening, everyone. So we're about to start. Yesterday we had day for yesterday we had completed our discussion on the properties of the nucleic acids. We talked about the genetic code. We had talked about mutation, and today we are going to start with the DNA synthesis. If time permits, we will go on to RNA and protein synthesis, but it is unlikely. We'll most probably do them tomorrow. So today we are going to be talking about the DNA synthesis, also known as replication in the prokaryotes. We are talking about the DNA synthesis as it is occurring in the prokaryotes. Once we have discussed the DNA synthesis in the prokaryotes, then we'll quickly compare it with the differences, minor or major, whatever is there, as seen in the eukaryotes. So first we'll do the DNA synthesis or the replication in prokaryotes and then quickly extrapolate what is seen in the eukaryotes. So remember, replication means DNA synthesis. Now, few things you will remember from our discussion of DNA. Number one, two strands of DNA will always be anti-parallel, and of course they are complementary. Now, first thing to understand is from where to start the synthesis of DNA and how to start the synthesis of a new DNA. So in this context, what you should know is the unique property of replication. Replication is semi-conservative. What this actually means, if you have the two strands of DNA, before you can replicate them, what we do, we separate the two strands of DNA. We separate the two strands of DNA, and each strand will now act as the template for the synthesis of a new strand. Each of the old strand will act as a template for synthesis of a new strand, and net result, you get two new DNA. You get two new DNA, but please note, in each of the new DNA, one of the strand is old. It is coming from the parent strand, and that is why we use the term semi-conservative replication. The replication is semi-conservative in nature. The replication is semi-conservative in nature means one of the old strands is conserved. Okay. So what we have to do as a first step, we have to separate the two strands of DNA so that we can carry out the replication. Now the question arises, from where are we going to separate the two strands? What is the region in the DNA from where the separation will occur and why? For this, we go back to the Chargaff rule. In the Chargaff rule, we had said A will pair with T. The G will pair with C. When we said this, at that time we did not specify the number of hydrogen bonds between A and T and G and C. But I'm sure you know that A and T will have 2 hydrogen bonds. The G and C will have 3 hydrogen bonds. And if you want to break the bonds, which one is easier to break? Is it easier to break 2 hydrogen bonds or is it easier to break 3 hydrogen bonds? Obviously, it is easier to break 2 hydrogen bonds, and this is what is used in the process of replication, when we want to separate the two strands of DNA, we look for an area which is AT rich. We look for an area which is AT rich, be lesser than the other strands. At this point, we allow the molecule called ORI binding protein to come and attach. Why do we call it ORI binding protein? Because this place from where the replication is going to start is known as origin. It is known as origin of replication, in short ORI. Origin of replication, in short we call it the ORI, and therefore the protein which will come and attach here is known as the ORI binding protein. So the ORI binding protein will come and it will attach, to the origin of replication, or the ORI. A very important point to note is when I say ORI, it includes both the strands. ORI always includes both the strands. Alright, ORI does not mean just one strand, it includes both the strands. So one ORI is two strands of DNA. Two strands do not make up two ORI. When I say one ORI, it includes the two strands of the DNA. It includes the two strands of DNA. So first there is identification of the ORI, and at ORI there is attachment of ORI binding protein, also known as ORI response elements or the ORI binding elements. Any of these terms can be used. The ORI binding element, the ORI binding protein, or the ORI response element, they come and attach at the AT rich region, and what they will do, they will cause separation of the two strands of DNA by breaking the hydrogen bond. So in the next step you find the two strands are separated in the ORI region. In the ORI region, the two strands are separated, but don't forget that the two strands are complementary in nature, and therefore after some time they will try and rejoin together. But we don't want this to happen, so we prevent it by bringing in another set of protein which will prevent the two strands from joining. Another set of proteins come in and they sit in between the two strands and they prevent the two strands from joining. These are single strand binding proteins. These are the single strand binding proteins, prevent premature re-annealing of the separated strands at ORI. They prevent the premature re-annealing of separated strands of ORI. So now the ORI has been separated, the separation has been stabilized. Now it is stable, it will stay there and it will allow the other enzymes to act. So at this point, an enzyme which is able to carry out further separation of the DNA strands comes in, and that is helicase. Please note, helicase has the unwinding activity, means it breaks the hydrogen bonds irrespective of the number, whether it is 2 bonds or 3 bonds. It is able to break the hydrogen bonds between the nitrogen bases and separate the two strands of DNA further. A very important thing to note is helicase will work simultaneously in both the directions. So a pair of helicase will come in. You can see there are two proteins here, a pair of helicases will come in and they will start separating the two strands in opposite directions simultaneously from the ori. From the ori separately, two helicases will go in the opposite directions and they will cause the separation of the two strands of DNA simultaneously. This is the helicase. So next step, what do you find? The two strands of DNA are separated to a significant degree, and after the separation there is DNA polymerase. In this case it is the DNA polymerase 3, and the second is what is known as primase. So the synthesis of new DNA occurs, but because the two strands are anti-parallel, the synthesis pattern is not the same. We'll discuss what is happening here. Remember, this was 5 dash to 3 dash, 5 dash to 3 dash. Also recall that synthesis of new nucleic acid will always occur in 5 dash to 3 dash direction, meaning this would be the 5 dash end and 3 dash end. This would be the 5 dash end, 3 dash end, 5 dash end, 3 dash end, 5 dash end, and 3 dash end. So let's first talk about the DNA polymerase a little bit. DNA polymerase has a very small but significant problem. Whether it is the eukaryotic DNA polymerase or it is the prokaryotic DNA polymerase, it has a very significant problem, and that problem manifests like this. It cannot, I'll repeat, it cannot initiate DNA synthesis. What do I mean by initiate? It cannot join two free deoxynucleotides. It cannot join the two free deoxynucleotides. So how will the replication start? Somewhere you have to start. Somewhere for the first time you would have to add the two nucleotides. Only then gradually we can increase the size of the chain. So somewhere you have to start. However, the DNA polymerase cannot initiate the DNA synthesis. So we need something else to initiate the synthesis. So we make a primer. Obviously, because DNA polymerase cannot initiate the synthesis, this primer must be RNA in nature, and the enzyme synthesizing the primer is known as primase. So we need a primase. The primase will synthesize a primer. The primer is called the primer is RNA in nature, and over that the DNA polymerase can act. To simplify the function, to remember what the DNA polymerase does and what it cannot do, I'll give this simple example. Suppose you request a caretaker to look after your babies. So the caretaker gives you some conditions before he will take care of your babies. Are your babies small? Means two free nucleotides. Here it is represented by two free nucleotides. You are asking him to join them. Are your babies small? You say yes. Then the DNA polymerase will say sorry, I cannot do that. I'll not take care of your babies. Two small babies is too much. Meaning the two small nucleotides have a lot of free energy which cannot be encountered by the DNA polymerase 3. It is unable to hold the two nucleotides together, so it cannot act. Then the caretaker asks you, okay, you have two children. Are both of them grown up? Meaning, are they two different nucleic acids? Are you asking me to join the two nucleic acids? And the reply will be no, I cannot join the two nucleic acids. So two things have come out. The DNA polymerase cannot attach free nucleotides. it cannot attach the free nucleotides. It also cannot attach the nucleic acids. Then what is the action of the DNA polymerase? The DNA polymerase will ask you, okay, do you have one big child and one small child? Say yes. Then he says, okay, I'll work for you because I can make use of the older one to take care of the smaller one. So basically the older one will be stable, it is a bigger molecule, it will be stable, and to this the free one can be captured and attached. So what is happening here? Nucleic acid to free nucleotide is attached. The function of DNA polymerase is it will only attach a nucleotide to a nucleic acid, not to another free nucleotide. It will not attach the two nucleic acid. So this is what is happening here. I told you that the primase will synthesize a primer. This primer, although it is RNA in nature, it is still a nucleic acid, and on this primer now we will build the DNA by the action of DNA polymerase. So if you look very carefully at this segmented fragment on the second strand, you find in the beginning you have the RNA, that is the primer, and after that you have the DNA. So the primer is extended to give us the DNA. Because the two strands are antiparallel, obviously the synthesis will occur in opposite direction, because the synthesis always occurs in 5' to 3' direction. Because of this peculiar pattern of synthesis, one of the strands is called the leading strand. Why it is called the leading strand? Because here you find that a continuous synthesis has occurred. A continuous strand is being synthesized. The second strand is commonly known as the lagging strand. And obviously on one hand where we have the continuous synthesis, where we have the discontinuous synthesis. Here we have the discontinuous synthesis. Another important point to remember, this fragment containing both RNA and DNA is known as Okazaki fragments. Please remember, the Okazaki fragments will contain both. They will contain both DNA and RNA. They will contain both DNA and RNA. So the Okazaki fragments contain both DNA and RNA. They are present on the lagging strand where the discontinuous synthesis occurs. Now when you get the final DNA, we don't get the small fragments. The Okazaki fragments are not there, so a phenomena must be occurring whereby these strands are joined together. So let's see what happens to join these strands together. There is entry of another DNA polymerase here, and this one is commonly known as DNA Polymerase One. So what the DNA Polymerase One does, it will come and overlap the area where there is gap between the nucleic acids, and then it will carry out its magic. What is the action of DNA Polymerase One? So this is the lagging strand, and here we had seen the primer is there. I'll draw it like this. And beyond that the DNA is there. First the primer is synthesized and then the DNA is synthesized, but remember at the core both of them are nucleic acid. Similarly we have another primer, and beyond that another nucleic acid. Okay, so the DNA polymerase 1 will come over this area where the RNA meets the DNA, where RNA comes over and meets the DNA. This is where it will come in. Remember, the orientation of the second strand is 5 dash to 3 dash, 5 dash to 3 dash. Now, DNA polymerase has a very important and peculiar function. Particularly the DNA polymerase 1, it has what is known as 5 dash to 3 dash exonuclease activity, also known as the proof reading activity, because if there is any mistake in the replication, it can be corrected by the exonuclease. So we have the 5 dash to 3 dash exonuclease activity in addition to the polymerization activity. So what this exonuclease does, it starts removing the RNA from the 5 dash end. See here, DNA should be there, but what we find is the RNA. So it will remove the RNA and replace the RNA by a nucleotide. See, at the point in time, the nucleotide is not attached to any of them. Okay, it is not attached to any of them. But if you want to attach it, where should you attach it? To this Okazaki fragment or to the second one? To which Okazaki fragment are you going to attach the nucleotide? The answer is very simple to this question. The free nucleotide can only be attached at the 3 dash end. It cannot be attached the 5 dash end. We had said when you're talking about the nucleic acid, the free nucleotide will always get attached at the 3 dash end and not at the 5 dash end. So we have removed the nucleotide from the 5 dash end of the primer, but we cannot replace the nucleotide at 5 dash end, means it must go back to the 3 dash end, means to the second nucleotide. And this is how one by one the ribonucleic acid are replaced by the deoxyribonucleic acid. But herein what you notice a very important phenomena: whatever primer has been replaced by the deoxynucleotide is added to the next Okazaki fragment, so that at the end you are left with the two deoxyribonucleotides sitting close by but not joined together. Remember, the two nucleic acid cannot be joined together by the polymerase. The polymerase will not be able to join together the two nucleic acid. So 5 dash to 3 dash exonuclease activity will remove the RNA primer. DNA polymerase will subsequently fill in the gap, but for the joining purpose, we have the enzyme DNA ligase. So DNA ligase will come in and it will join the two Okazaki fragments together. So here two things are happening. First, the primer is replaced by, and the answer is DNA polymerase 1. Second, the two Okazaki fragments are joined together by, the answer will be the DNA ligase. The answer will be DNA ligase. So if you look at this structure, what is happening here? We have two nucleic acids getting synthesized on opposite strands. One is the leading strand where the continuous synthesis occurs. The second is the lagging strand where discontinuous synthesis occurs. The fragments here which you can see are known as Okazaki fragments. Okay. So gradually all the DNA will be copied. Each of the strands of the DNA will be copied and will reach the end of the DNA. We will reach the end of the DNA. Before reaching the end of the DNA, one very important point which I want to highlight here, and that is something like this. The helicase has a very important side effect. The helicase has a very important side effect. What it does, it causes unwinding obviously, but besides unwinding it also causes what is known as torsional strain, and if you don't rectify the torsional strain is going to result in positive supercoiling. So you must remove the torsional strain so that positive supercoiling does not occur. Once the positive supercoiling sets in, then it becomes very difficult to release it from the supercoiling and then separate the two strands of DNA. So we have to relieve the torsional strain. The torsional strain is relieved by a group of enzyme known as a topoisomerase. Very briefly, the topoisomerase relieve torsional strain by introducing a cut releasing extra turns. During unwinding, extra turns are introduced, so the extra turns are released, and after that the DNA is rejoined. So topoisomerase will relieve the torsional strain by decreasing the number of turns which are a result of the torsional strain. Here you should remember the topoisomerase can be of two types, rather two classes, class 1 and class 2. The class 1 decreases torsional strain by 1 turn. Class 2 decreases torsional strain by 2 turns. Okay, so torsional strain decreased by 1 turn by topoisomerase 1. The torsional strain decreased by 2 turns by topoisomerase class 2. Among the list of class 2 topoisomerase, we have a very important topoisomerase. This is known as DNA gyrase, found in bacteria. It is found in bacteria and it is very important. The topoisomerase 1 is enough for eukaryotic replication. It is slow, but still it is enough for eukaryotic replication, whereas the class 2 is required for the prokaryotic replication. What I want to say here is, I am saying that DNA polymerase 1 is enough for eukaryotic replication. This implies that it is also present in the prokaryotes. I'm saying that 2 is required for prokaryotic replication, but at the same time the 2 is also present in the eukaryotes. So here we are talking about the preference. I'm not saying where the polymerase 1 is present or the 2 is topoisomerase 1 or 2 are present, but rather I'm talking about what is the requirement. The topoisomerase 1 is enough for the eukaryotes, whereas topoisomerase 2 is critical for the prokaryotes because they are replicating at a much faster rate. They are replicating at a much faster rate. So I'll do a quick recap. The helicase causes unwinding along with torsional strain. If not relieved, the torsional strain will cause the positive supercoiling, after which the further replication cannot occur. So the torsional strain is removed by what is known as the topoisomerase class of enzymes. The topoisomerase relieve torsional strain by introducing a cut in the DNA. After that, they release the extra turns and they cause rejoining of the DNA strands. There are two classes of the topoisomerase, the class 1 and the class 2. Class 1 decrease the torsional strain by 1 turn. The class 2 decrease torsional strain by 2 turns. And a classical example of DNA gyrase of type 2 is DNA gyrase in the bacteria. You will have read in your pharma that DNA gyrase can be inhibited by the fluoroquinolones. It can be inhibited by the fluoroquinolones, for example, the ciprofloxacin. So the DNA gyrase can be a bacteria for, sorry, it can be a target for the antibiotics. Okay, so we said the separation of the two strands occurs, then the primer is synthesized, then it is extended by the DNA polymerase. Now, two very important things. First, coming to the previous original structure. See, if you look at this structure, which has appeared due to separation of the two strands, this is looking like a bubble. Further, if you look at the second strand, this becomes bigger in size, so it is an expanding structure like a bubble. And because here the replication occurs, we call it the replication bubble. So what you're looking at is the replication bubble. Now, one very important thing to note, remember the helicase was traveling simultaneously in both the directions, right? So in the opposite direction, whatever was the lagging strand on left side will become the leading strand on the right side. Similarly, what was on the right side, a leading strand will become a lagging strand on the left side. So what do you see? We find that replication occurring simultaneously in both directions, which was the replication bubble. So if you look at one half of this picture, it is comparable to a fork. You can call it a fork in the road or the fork that you eat. It is comparable to a fork, only one half. When you look at only one half, and therefore just one half, just the one half of this replication bubble, just the one half of this replication bubble is commonly known as the replication fork. It is commonly known as replication fork. So two replication fork together make up one replication bubble. Two replication fork together make up the one replication bubble. So remember these terms, the replication fork and the replication bubble. We reach the end of the replication on one of the lagging strands. So at the end what you will find? At the end what you will find? The Okazaki fragments. The Okazaki fragments. So recall what happens when we have the role of the DNA polymerase 1. The RNA part of the Okazaki fragment is removed, and the gap is filled by extending the next Okazaki fragment. So this Okazaki fragment will be used for extending and filling the gap, and subsequently the two will be joined by the action of DNA ligase. But what happens when you come to the last Okazaki fragment? This Okazaki fragment after this, if you remove the primer, the gap cannot be filled by any other mechanism, but the RNA is unstable. After some time it will automatically disappear. So what do you do now? How are you going to fill the gap left behind by the RNA primer? The answer is this gap cannot be filled by DNA, for the very simple reason the fresh DNA cannot be synthesized by the DNA polymerase. It must be added upon pre-existing nucleic acid, which is normally the iron, and there are no other nucleic acid beyond the end of the DNA. So this part will remain empty, and this is why we had mentioned right in the beginning of the DNA, one strand is smaller than second strand. When we are doing the telomere, I had said that one strand of DNA is smaller than second strand at end of DNA. At the end of DNA, you will always find that one strand is smaller than the second strand. This is because the gap occupied by the last primer cannot be replaced. The gap occupied by the last primer cannot be replaced, and it will always bear this gap. So this is what is known as telomere shortening. This is what is known as telomere shortening. If you recall, when we are talking about telomere, then I had given you a very important information. The information was the telomere protects the ends of DNA, and it protects them from the DNA repair machinery. So now what is happening? The telomere shortening is occurring. After some time, there will be complete absence of the telomere. All of it will gradually be lost by the shortening after each metabolic cycle. Once this happens, the DNA will become unstable. They will try to join together. They will become unstable when they join together. After that, the apoptosis will occur. So the telomere exhaustion is a marker of how many times a cell can divide. So telomere exhaustion will tell us about the number of times a cell can divide, and this is known as the Helix limit. This is known as the Helix limit. For humans, it is in the range of approximately 75 divisions. Rather, I would be saying it is closer to 70 divisions. It can be 70 to 75 divisions. So this is the number of times you can say somatic cell can divide. After that, it is likely to undergo apoptosis. Obviously, there can be some variation, can be 75, it can be 65, but on an average, after 70 divisions, further ability to divide is lost. If it is attempted, apoptosis will occur. Now, this is very interesting to say. We say that there is Helix limit. Helix limit says only 70 divisions are required, but I'll tell you certain problems. For example, if you look at your head, not at my head, you'll find a lot of hair, and this hair keeps growing. It keeps growing, it keeps growing, it keeps growing, and maybe in a month it grows up to 10 centimeters. That's barely 4 inches. So it grows to 2 to 4 inches in a month. It will vary from individual to individual. Now if you look at the cells which make up the hair, you'll find the hair cells are very small in size. They are barely 20 microns in height. If you look at their height is barely 20 microns, meaning to reach a height of 10 centimeters you will need roughly, I'm saying roughly, 5,000 divisions. See, 20 microns is one cell. So if you reach 1000 micron, that will be 50 cells. 50 cells stacked one over the other will be 1000 microns, and that is 1 millimeter. So from 1 millimeter, if you go to 10,000 microns, that is 1 centimeter, that will be 500 cells. And if you go to finally 10 centimeter, it will be 5,000 cells. So this means in one single month the hair follicle divides 5,000 times, and I was just saying that the Helix limit is only 70 divisions. So there must be a mechanism to bypass the Helix limit. How are we going to bypass the Helix limit so that unlimited number of divisions can occur in those cells where the cell division is required? And that is by the enzyme called telomerase. We have an enzyme, rather it is an enzyme complex, which is the telomerase, whose primary function is the regeneration of telomere. So the telomere can be regenerated by the action of telomerase, and because there are certain cells which require unlimited number of divisions, the telomerase will be super active in those cells. Okay, telomerase will be super active in those cells, A few points about telomerase. There are two primary components, although there are very large number of proteins, but two of them are primary components. One is the template RNA. Template RNA. This template RNA is also commonly known as telomerase RNA copy. Telomerase RNA copy. TERC. TERC is the telomerase RNA copy. And the second is the exotic reverse transcriptase. Because it is present in the telomere, we call it the telomerase reverse transcriptase. It's present in the telomerase, so we call it the telomerase reverse transcriptase. Obviously, there are very large number of proteins besides these two, but we are primarily concerned with these two. The template RNA or TERC is complementary. It is complementary to the telomere sequence, and if you remember, the telomere sequence is very specific. It is self-complementary repeating segment. So what is happening now? See, this was the end of DNA as we had seen it. So here comes the telomerase complex. The telomerase complex will come in, and it brings along with it the TERC, telomerase RNA copy, which is complementary to the telomere. Therefore what happens, it is able to form the bonds with the extending overhanging edge of DNA. Alright, and a part of it is extending beyond. Now the overhanging edge belongs to the RNA, and remember, this lagging was happening at the 3' end. The lagging was happening at the 3' end. So now what we can do, using the enzyme reverse transcriptase. What does reverse transcriptase do? Normally the polymerase or transcription, what it does, it converts the DNA into RNA. When we say reverse transcriptase, we will convert the RNA back into DNA. So looking at the RNA sequence, we write the complementary DNA nucleotides and extend the DNA at the 3' end. Once this extension is completed, the telomerase will disappear. The telomerase will disappear. As you can see, they have almost disappeared. Okay, right? And after that, they come and join to the new overhanging edge again. They come and join to the new overhanging edge again. They'll further extend it by certain nucleotides, again disappear, then further extend it by a few nucleotides, and again disappear. So typically about 3 to 5 times the end of nucleic acid is generated when the telomerase comes in the picture. If it is not there, it is not there. If it is there in the picture, 3 to 5 times it will increase the end of the DNA. Once it is completed, either in the same cycle or in the next cycle, the new extended DNA can again participate in the replication. So please note, the previous DNA was at the arrow that I have marked, and therefore the new DNA which was synthesized was a length shorter than the old DNA which was longer. But now when you have extended the lower DNA strand, the new endpoint for the new DNA is way longer than the initial endpoint for the old DNA. although it is still shorter than the new end of DNA, but in comparison to the old strand, it has become longer. So because it has become longer, it will be able to survive for several more divisions even if the telomerase does not come into the picture. And this is how the telomerase will regenerate the telomere. Once the telomere is regenerated, those cells can divide unlimited number of times, because now the Helix limit will not be hit. It is like playing hide and seek. I tell you to go and count backwards. So the person starts counting backwards: 70, 69, 68, 67, 66, and then you say dhappa. Start counting again back from 70, not from 70, start counting from 75. So start counting from 75, 75, 74, 73, 72, 71, 70, 69, and you say dhappa. Again start counting, but not from 75, this time from 80. So every time the telomerase acts, it increases the length of telomere so that more telomere is available to be lost during each division, and the number of possible divisions increase. If the telomerase is continuously active, the telomere will keep getting longer in size and more number of cell divisions can occur. So this is the importance of the telomerase. Importance of telomerase: it is able to regenerate. We had said it is able to carry out regeneration of the telomere, and the tissues where it is active. Please note down a free tissues where the telomerase is active. The telomerase is active in, I told you, the hair follicle. It is active in hematopoietic stem cells. It is present in hematopoietic stem cells. It is obviously present in the endothelium of skin and GIT. They are continuously growing. They are continuously growing inside the body. They are shed off, new skin comes, and then they again shed off and new skin comes. So endothelium of skin and GIT. And a very important name in this list is the telomerase are active in about 50 to 60% of malignancies. This is a newly identified feature that a large number of cancer cells do not undergo apoptosis, whereas they should have undergone apoptosis due to depletion of telomere. Now, some of them, about 50% of them, do not have the active apoptotic cascade. Either P53 is not there, or P21 is not there, or the GAD is not there. So, some or the other factors about the apoptotic cascade are missing, and therefore apoptosis cannot occur in 50% of cells. The remaining 50% of cells, apoptosis is normal, and still the process does not occur. Why? So, there we find that the telomerase has been reactivated, and it is not allowing the telomere to get exhausted. If the telomere does not get exhausted, we don't need to use the anti-apoptotic cascade. Alright, so these are some examples. If you remember, the telomerase is active in the hair follicle, hematopoietic stem cell, the endothelium skin and GIT, and about 50 to 60% of the malignancies. So this brings us to the end of the replication. So few things I like to summarize here. First, I like to summarize the proteins which have been used in the process of the replication. We start off with the ORI binding protein. What do the ORI binding protein do? They cause separation in the origin of replication, commonly known as ORI. Once the two strands of DNA are separated, then in the picture comes single strand binding protein. What they do? They prevent premature re-annealing. They prevent premature re-annealing. Following the single strand binding protein are the class of enzymes topoisomerase. The primary function of topoisomerase is to prevent the topoisomerase prevent, or you can say relieve, torsional strain. They relieve the torsional strain. After the action of topoisomerase, we have the very important primase. The primase synthesizes RNA primer. This means the primase must be an RNA polymerase. The primer is then extended by the action of the DNA polymerase. DNA polymerase has certain important functions. It polymerizes DNA and it also carries out proofreading. It also carries out the proofreading of the replicated DNA. It carries out the proofreading of the replicated DNA. In addition to the main polymerase, which is the type 3, we also have the DNA polymerase type 1. DNA polymerase type 1 again has very specific function. What it does, it removes the primer and replaces it with deoxynucleotides. It is replaced by the deoxynucleotide. DNA polymerase 3 will mainly cause polymerization of DNA and the proofreading, whereas DNA polymerase 1 will primarily cause the removal of primer and replacing it by the deoxynucleotide. And the last one in the list, can you guess? It is the DNA ligase. It is the DNA ligase. What the DNA ligase does, it joins two nucleic acids. It will join the two nucleic acids. So we summarize the action of all the proteins. The ORI binding protein will cause separation in the ORI region. The single strand binding protein will join with the separated strands and will prevent the premature re-annealing. The topoisomerase at the same time will act and they will relieve the torsional strain. After that, the primase will synthesize the RNA primer, which is extended by the DNA polymerase type 3. The DNA polymerase type 3 will primarily carry out the polymerization of the DNA. The DNA polymerase 1 will remove the primer and replace it with the deoxynucleotide. Once all the primer has been removed, the DNA ligase will join the two nucleic acids, rather it will join the two Okazaki fragments. It will join the two Okazaki fragments. So this is the primary function of all the different proteins which are involved in the replication. What we'll do, we'll quickly take a look at the process again to recap what exactly is happening when we are talking about the replication. First we have to identify the ORI. Which is the origin of replication. The ORI is AT rich segment on the DNA. Two strands of DNA make up one ORI. At this ORI, there is attachment of the ORI response element, the ORI binding element, or the ORI binding protein. They'll come and attach and they'll cause separation of the two strands of DNA. Once the two strands of DNA are separated, their separation is stabilized by the action of the single strand binding protein, by the separation of single strand binding protein. And after that, there is entry of the helicase, which will cause the further separation of the two strands. Two helicases come in and they start separation of the two strands simultaneously in the opposite directions. Following this separation, there is entry of the primase. Which will first synthesize the primer, and the primase is then extended by the action of DNA polymerase. The primary DNA polymerase is the DNA polymerase 3. Because the two strands are antiparallel and the nucleic acid is always synthesized in 5' to 3' direction, we find a different pattern of replication on the two strands. On one strand we find the continuous synthesis, we call it the leading strand. On the second strand, we find the discontinuous synthesis, we call it the lagging strand. The lagging strand has Okazaki fragments which contain both DNA and RNA. Now, to join together the Okazaki fragments, we need the DNA polymerase 1. The DNA polymerase 1, what it does, the DNA polymerase 1, by the 5' to 3' exonuclease activity, it will first remove the primer. RNA primer is removed and it is replaced by the deoxynucleotide. Once all the primer has been removed, the two Okazaki fragments have to be joined. The joining is done by the action of a DNA ligase. Once this is done, we have the continuous DNA. In this context, I told you that we get the replication bubble. Replication bubble is the whole segment which is getting opened up by the action of helicase, and subsequently it is replicated by the action of the polymerase. That's why it's a bubble where replication occurs. So the replication bubble. And if you look at one part of this replication bubble, what you get to see is the replication fork. This structure resembles a fork, so we call it the replication fork. A fork where replication is occurring is the replication fork. A small problem comes when we reach the end of DNA. The space occupied by the last primer cannot be filled, and this results in shortening of the telomere. This results in shortening of telomere, because of which after a certain number of divisions the cell cannot divide further. This number of divisions is called Helix limit. For humans it happens to be in the range of 70 divisions. However, there are certain cells where the unlimited cell division is required. So there we have the enzyme complex called the telomerase. Telomerase carries out the regeneration of telomere. The two important components of the telomerase are the telomerase RNA copy, that is the template RNA which is complementary to the telomere, and the telomerase reverse transcriptase which will use the RNA as template and make the DNA opposite of the transcription. The result of the telomerase, we said there is a regeneration of telomere. The regeneration of telomere ensures that unlimited number of divisions can keep occurring. Some tissues where telomerase is majorly active include the hair follicle, the hematopoietic stem cells, the endothelium of the skin, and GIT. And in addition to that, about 50 to 60% of the malignancies also show activated telomerase, whereby they can bypass the apoptosis. Once the replication is completed, please remember the DNA must rearrange as chromatin. Why? Because replication is occurring in the S phase, and if you recall, only in the M phase, the mitotic phase, we have the chromosome. In all other phases we have the chromatin. This means in the G0 phase, in the G1 phase, in the S phase, and the G2 phase, we'll get the chromatin. Only in the M phase we are going to get chromosome. Okay, so this is about the DNA replication. Few things which are going to cover tomorrow is number one, difference between prokaryotic and eukaryotic DNA polymerase. This is very important, very frequently asked. Number two, DNA damage and the DNA repair machinery. Number three, I'll show you the differences between prokaryotic and the eukaryotic replication. Number four, I'll mention some drugs which can inhibit the DNA replication. Although we have already mentioned some of them, but I'll again list them, some drugs which can inhibit the DNA replication. After that, we'll go on to the transcription and then the translation. We'll finish it off with certain regulatory components and certain techniques. For example, the polymerase chain reaction, the RT-PCR, a little bit about the fish, if time permits, about ELISA, and certain other small important techniques will be mentioned. We can also talk about the techniques to separate the proteins tomorrow. There was some confusion today, because of which we could not start at 8 o'clock, but we'll coordinate well tomorrow so that we get a clean about 2.5 hours. That is all is required to finish our discussion on biochemistry. If you have any questions, please let me know, else we will call it good night now and meet again tomorrow. Any questions? Pooja, Vinod, Manisha, Dharvat, any questions? Please let me know. So if there are no questions, I think we can call it good night today, and we'll finish the biochemistry tomorrow. So if you have any other questions or any topic you want to ask, uh, tomorrow's timing will inform you right in the morning. In the morning, we'll coordinate with Dr. Manoj and Dr. Shravan and update you before 10 o'clock. We'll update you before 10 o'clock what is the time, or maybe we can decide tonight and message you what will be the time for tomorrow's